Hello, welcome to the session all about SCA4. It is my absolute pleasure to introduce our speaker today, Dr. Stefan Pulst, who is the chair of the Department of Neurology and professor of neurology and human genetics at the University of Utah in Salt Lake City. He holds the Rinsbacher Endowed Chair for Neurodegeneration Research. He trained in Hannover, Germany, and at the Harvard Longwood Program in Boston. Prior to moving to the University of Utah in 2007, he was professor of medicine, neurology, and neurobiology at the UCLA School of Medicine and neurology chair at the Cedars-Sinai Medical Center. Researchers in the Pulse Laboratory focus on identifying human disease genes and subsequent modeling of diseases in cells and genetically engineered mice. His laboratory works at the interface of discovering disease genes and molecular targets and developing gene-targeted therapies. His basic science interests are the role of RNA-binding proteins in autophagy, cellular stress response, and the translation group in his laboratory extends these insights to targeting specific genes using antisex oligonucleotides, siRNAs, and viral gene therapy. Dr. Pulst has authored over 300 research papers, reviews, book chapters, and edited or co-edited 5 books. The latest one on trials for cerebellar ataxias from cellular models to human therapies. He lectures nationally and internationally and advises research centers and universities on clinical and research strategies. Dr. Pulst was awarded the Kotsias Prize by the American Academy of Neurology and the Singhal Prize by the World Federation of Neurology, and has received an 8-year R35 Merit Award from the National Institute of Neurological Disorders and Stroke. He became an elected fellow of the American Association of the Advancement of Science in 2021 and currently serves as the co-chair of the NINDS Board of Scientific Counselors. Dr. Pulst is also the founding editor emeritus of the Neurology Genetics Journal. And with that, Dr. Pulst, the floor is yours.
Thanks, Celeste. Thanks for the introduction and thanks for inviting me. You heard the acronym NIH, NINDS several times in the introduction, National Institutes of Health and the National Institute of Neurological Disorders and Stroke. And I will spend just about 2 minutes on informing you what is happening at the NIH. I've been funded by the NIH for almost 40 years now. We are seeing unprecedented dismantling of the NIH research structure and the funding. Several awards that directly affect patient care, development of therapies have been canceled. What you have read in the newspapers about so-called indirect costs that I just thought to be bureaucratic costs are actually absolutely necessary for a research enterprise. They pay the mortgages on our buildings, keep the lights on. They do important things like support the institutional review board that makes sure that patient safety is assured and that human subjects are properly treated in trials. They do things like support instruments. They make sure that our animals that we use for research are treated well. So very important things. This is not bureaucratic waste. It is an essential part of the research enterprise. We're also seeing that grants are being canceled. One at the University of Utah was just canceled, a very large grant that reached out to underserved populations. And in Utah, we are a rural state. We are surrounded by rural states. That meant that we are thinking of ways to extend research to individuals who live far away from our main academic center. So if you're interested in finding cures for brain diseases, as my department is dedicated to, reach out to your representatives and senators and tell them how important research is for you in all regards. Funded by the Department of Defense, by the National Institutes of Health, by the CDC. These are jewels in our crown. I think my European colleagues are always very envious about the support that we are receiving in the United States. So having said this, let's talk about SCA4, one of the newest, maybe the newest SCA that has been discovered. And I will take you hopefully through an interesting journal, both historically and genetically, that led to the discovery of this new gene. So what are my takeaway points? SCA4 is an autosomal dominant ataxia. In contrast to other SCAs, it has taken 30 years of identifying this gene because it was in a part of the genome that was very difficult to decipher. In contrast to other SCAs, the ataxia is mixed. It has two major components, one in the cerebellum and one in the peripheral nerves. And I will talk about this a little bit more. Like many other SCAs, it is a ataxia that is caused by expansion of a DNA repeat. But in contrast to what you may have heard, polyglutamine ataxias or CAG repeat ataxias, the repeat is made of a different DNA repeat. And as far as we know currently, this mutation may have happened only once in human history in a person living in southern Sweden. We are at the beginning of unraveling this, but so far everything points to Sweden and especially southern Sweden. I will talk to you about a study that my group led, but it wouldn't have been possible without the collaboration, input, and ideas and excellence of many people, especially in Germany and in Tübingen, led there by my colleague and long-term friend Olaf Ries, who is professor and chair of the Human Genetics Department. So just we all may have slightly different backgrounds to catch everybody up. What are SCAs? They're actually very well defined. The SCA stands for Spinocerebellar Ataxia. These are neurodegenerative disorders that primarily affect the cerebellum, and in particular, a prominent cell in the cerebellum, they're called the Purkinje cell. They're autosomal dominant. That means that transmission of the genetic variant occurs vertically from parent to child. It also unfortunately means that 50% of children are at risk statistically to inherit this variant. But has to recognize that in the addition to the cerebellum, often other parts of the nervous system are involved as well. And we now have 50 plus SCAs, and they're all caused by different genes that very often have very little in common. So a brief history of the SCAs. SCA1 is named 1 because it was the first SCA mapped to a chromosome in 1977. And it took several years. It was at the beginning of the genetic revolution in 1993. The laboratories of Harry Orr and Huda Zogby identified the genetic mutation. It was a DNA repeat mutation resulting in the expansion of a polyglutamine tract. I will talk about this in a second. Then we discovered SCA2 in 1996 after it had been mapped to 1993, and so on. You can see the numerals actually tell you when a SCA was mapped to a chromosome. So SCA4 was already mapped in 1996, and identification of the actual mutation and the disease gene had to wait a long time. And that happened in 2024 when my group and 3 other groups actually identified this mutation, aided by the development of new sequencing technology. So let's spend 1 minute to catch people up who are not geneticists. Just to clarify certain terms, DNA provides the blueprint to construct proteins. Really think of it as a contractor or an architect giving you a blueprint how to build a house. The building blocks of the house are proteins, and the bricks of the walls are really amino acids. Their incorporation into a protein is detailed in the blueprint by 3 DNA bases. So 3 DNA bases basically spell out which amino acid gets incorporated. There are 4 DNA bases, A, T, C, and G. Let me give you an example. If 3 letters are CAG, that specifies that a glutamine is incorporated into the protein. We unfortunately also have single letter codes for these glutamines to make communication easier. Q stands for glutamine. So poly-Q disorders is the same as polyglutamine disorders. For some amino acids, several codons can specify the incorporation of that amino acids. For example, CAA also codes for glutamine. GGC, on the other hand, codes for glycine. Single letter code is G. So a polyglycine disorder is also a poly-G disorder. And glycine is specified by 4 DNA codons. Most of the repeats are actually imperfect. For example, as I showed there on the left side, I can point to it here. We have a stretch of CAG, CAG, CAA, CAG. So this is an imperfect DNA repeat, but it's a perfect polyglutamine repeat. Same here below for GGC. It's an imperfect repeat, interrupted, but it spells for a perfect polyglutamine repeat. Why is this important? We think that these interruptions actually stabilize the repeat. They prevent it from expanding further. And I will show you that in SCA4, just like in other repeat disorders, an imperfect repeat through mutations was made into a perfect repeat, and then we think likely expanded from there. Just to show, this is another example. This is the Huntington gene, also a polyglutamine disorder. Here you see the Huntington protein. It has a polyglutamine repeat in it in individuals that do not have Huntington's disease. And then when this repeat expands at the DNA level, codes for a longer glutamine repeat, disease develops. Another little thing to add is that proteins are encoded by a blueprint, but think of a contractor who gets little side notes of, this wall may be blue or red, or the homeowners hasn't decided yet. There are things that actually specify where a wall goes, and then there are these side notes. And what the cellular machinery does is connect these walls and takes out these introns. These introns are not without function. They have, as I said, certain side notes involved. Why am I telling you this? Because when you look at repeat diseases, the repeats can occur both in exons or in introns. And you can see here, we won't go through all these diseases, obviously, in detail, but there are a lot of neurological disorders that are caused by expansion of DNA repeats. The repeats may be in different parts of the gene. SCA4 is actually in an exon, so in the direct part of the blueprint that specifies directly what kind of protein is being made. But some of the recent ataxias that have been identified, SCA27B, that is somewhere here, and canvas, the repeat expansions occur in an intron, and of course also in the recessive ataxia called Friedreich's disease. So repeat diseases are very important to understand neurological diseases, and especially the mechanisms, how they cause disease. So I told you that it has taken a long time to identify the SCA4 gene, and actually the story of SCA4 starts in Utah with, in my department before I became chair, actually in 1996, by 2 individuals who are still very active in genetics, Kevin Flanagan, who is now leading the Gene Therapy Institute at the Ohio State University, and Louis Tacek, who is a professor at UCSF. And they saw patients. You can see here is a pedigree, a family tree of SCA4. In black are individuals that are affected. And you can see through many generations, and actually they found 1, 2, 3, 4, 5 generations that they were able to trace back that had SCA4. You can see there is vertical transmission from parent to children. You have to realize not everybody who does not have the disease is shown here. So this family tree is contracted to focus on individuals that have the disease. You can also see some families have many children because this disease actually traces back to the Utah pioneers, the Mormon pioneers, who came into the Utah Valley in 1847. Kevin Flanagan and Louis Tacek did what is called genetic linkage analysis. They traced the mutation with linkage markers with chromosomal tags, and they found out that this disease maps to the long arm of human chromosome 16. And, in 1996, we would have all thought it will take us 3, maybe at most 5 years to identify the mutation, but it actually took almost 30 years. So, what are the findings of SCA4 as Kevin Flanagan and Louis Tutchek found? It had an onset in midlife. They got a hint that actually the disease seems to strike earlier and earlier in subsequent generations. That topic had just been developed and identified in repeat diseases in neurology. It's a phenomenon called anticipation. Most patients had unsteady gait, but they had also unsteady reaching movements. In addition, they had changes in sensation. For those of you who have been at a neurologist, a good neurology exam always, the neurologist takes out a tuning fork and asks you, do you feel it vibrating on your toes or your fingers? And that was decreased. And that is quite unusual. Not many SCAs do not have this. Um, there were also reflex changes. Again, the neurologist takes out a reflex hammer and taps your reflexes, and you see the muscle contract. And in many patients, actually, in their example, in 100% of patients, the jerks at the ankle were absent, and it extended to other tendon jerks as well. And in a quarter of the patients, even the reflexes in the arms were diminished or absent. There were also speaking abnormalities, weakness, a sign, Babinski sign when the neurologist scratches the soles of your feet and your toe shows an abnormal response. Some patients had eye findings, and on exam by an EMG, electromyogram, and nerve conduction studies, many patients that had it, many of them showed abnormalities as well. I wanted to explain briefly the mixed nature of the gait unsteadiness. Here is the cerebellum, and think of the cerebellum as something that doesn't actively initiate movements, but it makes sure that movements reach their target. So the way the cerebellum computes this is when your motor cortex up here decides to move an arm, it doesn't directly let the cerebellum know because it goes down to nerve cells here in the spinal cord that make your arm move, but it lets it in passing know that this movement is intended. The cerebellum then just computes really how to reach the target, but in order to do this, it needs information from your limbs that feed up and go into the cerebellum. So it constantly compares where's the target? Are we close to the target? Is there something in the way? Is the target moving? Is my hand carrying a heavy weight? The cerebellum makes sure that everything works out fine. So in SC4, in contrast to other diseases where only the cerebellum is not functioning correctly, the input from below is also impaired and faulty. And so it is a double hit to your coordination. This is a patient that I saw that actually one of the first patients with SCA4. And by the way, there is Patty, I'll show her as Patty, my lab manager, who is the first author on the paper. There's a younger version of myself, about almost now 15 years ago. And let's look at the gait another time. And what you see is gait is right-based, as we see in cerebellar disorders, but you can also see that the legs are shaken out a little bit more. And the reason is that the nerves do not give the signal back to the brain that the extremity has moved. And so you see an exaggerated stepping of the gait because the information doesn't get back in time and in the right degree of movement. So we then, here you see the pedigree by Dr. Flanagan. We had a patient who lives in Utah who actually collected the family pedigree, and you can see it is enormous. We have it on a papyrus roll. It has about 1,000 living and dead members in this family. And you can see it is probably one of the largest pedigrees of any neurological disorder in the world. Here you see all the members of the SCA4 team in my group. And the ancestry traces back to Mormon pioneers that were born at the turn of the century, and the century is the 18th to the 19th century. So the patriarch was born in 1799. So we thought, we have identified several SCA genes, it would be just, relatively straightforward. With such a large pedigree, you can actually normally limit the region where the mutation is located to very small areas. And in the meantime, since about 2000, 2005, the Genome Project had developed novel sequencing technologies, things you may have heard about: RES, whole exome sequencing, WGS, whole genome sequencing. You can also look at the intermediates specifying between the blueprint in the DNA and the protein called RNA. You can sequence this. And we thought it would be pretty straightforward to identify the gene. Well, that was not the case. And in retrospect, or Patty actually identified the causative mutation, but I discarded it. For several reasons, we could not verify that there was an expanded repeat because the technology called PCR, polymerase chain reaction, didn't work for the repeat. And we now know why this is. The repeat has a very unusual composition that makes PCR very difficult to get to this long repeat. The gene in which the repeat resided at that point was not thought to be highly expressed in adult brain. As I will show you in the next slide, the repeat was interrupted not once, not twice, but 4 times. So likely stabilized and not prone to expansion. And when you think, well, why didn't you focus on something obvious? We now know that this region where the SCA4 repeat is located right here is full of repeats. So it would have, there were not only one repeat, there were almost 100 repeats that were just as likely to be mutated. Well, what came to the rescue was a novel sequencing technology called long-read single-strand DNA sequencing. I don't want to go into it in detail, but just imagine that you have a DNA reader and you can feed into this reader very long stretches of single DNA. So importance on single and long. Normally, the read lengths of DNA we can read is 150 DNA bases, or maybe 200, maybe 300. But in this new reading technology, we can do it to up to 10,000 bases. And it was really my friendship with Olaf Ries, whom I meet every year because I advise the University of Tübingen. He said, well, we have this new technology working. Do you want to try SCA4? And we sent him DNA samples, and all funded by the NIH, by the way. I have to really emphasize this. And he called me up in June of 2023 and said, I think we found the mutation. And what he had found is that there was, in all patients that had SCA4, there was a repeat, a GGC repeat, that was much longer in patients than it was in control patients. So this is about a repeat length of between 20 and 22, and all the individuals with SCA4 in our Utah pedigree and subsequently in some patients in Germany had an expanded GGC repeat. So we found it initially in our large Utah family. Subsequently, we have found the repeat also expanded in a family from Georgia, that is the US state of Georgia, 7 additional families in Germany, and a family from California. What all these families in retrospect have in common, that they trace their ancestry directly back to Sweden, especially to southern Sweden, or to northern Germany. So, very, very close to southern Sweden. So let me show you what this repeat does. The repeat is in a gene that I have to say I've never really heard of. It's actually, in the end, not important what this gene does. It is a factor that regulates gene expression, especially in early development of animal and human brains. It has very low expression in the adult brain, even in retrospect now. Here is the repeat in this gene. It's attached to one of the exons, one of the coding regions of a gene. And you can see the GGC repeat is 4 times interrupted. That's why I thought you have to convert 4 times, 4 individual events to make this into a perfect repeat. And I thought that was just unlikely. I was wrong. What happens when this repeat is translated in protein? There are many glycines. There's one protein interruption, further suggestion that this is likely not a disease gene, and then the glycine repeat continues. When we look at patient DNA, we see that now the repeat has become perfect. It is a GGC repeat, 42 times or more over. Here's the protein again, and now it has this very long glycine repeat that is attached to the end of the protein. There was something else that was very interesting. I told you I'd jumped ahead a little bit in the story that we think the ancestry of these patients traces back to southern Sweden. And what this long read sequencing also allowed us to look at the chromosome makeup near the mutation. And imagine that you could paint a chromosome with molecular DNA markers. And we did this, and we found that the chromosomes that contained the mutation all looked very similar in the vicinity of the mutation. And that tells you that they likely arise from one founding mutation. So as I said in the beginning, we now think that there was one mutational event in Sweden that gave rise to this repeat expansion. You can also see when we look at the other copy of the chromosome, I forgot to mention that we always have 2 copies, one inherited from the father, one from the mother. The other chromosome is different, but also in some patients very similar to the chromosome upon which the mutation arose. What that tells us that likely this mutation arose on a chromosomal makeup that was very common in southern Sweden. And in retrospect, it also explains why we had such a hard time to map this mutation by genetic linkage analysis. As I had mentioned, it was very difficult to verify the presence of the repeat with PCR, polymerase chain reaction, but we were able to do this finally looking at not at DNA, but at RNA in fibroblasts that we had isolated from patients, and subsequently with a lot of experimentation by Patty Figueroa, also in genomic DNA. It is nowadays possible to identify this repeat by PCR with very special conditions, but it is also possible to do it differently by using bioinformatic tools in regular short read whole genome DNA sequencing. So what do we know about this repeat in normal individuals? We were very lucky that Olaf Ries and colleagues had collected DNA and sequenced it from more than 6,000 individuals. So we could see there was a large number of individuals that did not have ataxia. Many of these patients were actually collected for cancer phenotypes. We can see the most common repeats are around 2021, but the repeat is variable in individuals without SCA4, a common story for all repeat disorders. Then there is an area where no repeats are found. So there's very little overlap. And when you get to 42 or more repeats in patients with SCA4, they had this repeat expansion. I'd mentioned to you that age of onset seems to be variable in this disease. And when we plot the repeat length versus the age of onset, we see the following. We see the longer the repeat, the earlier the onset. So here's the age of onset. You see when you have, 45 repeats, you have an onset of age 50 or 65. And at the other extreme, when you have 61 repeats, you have an onset around 12. We now have many more data points on this map, and it holds up with a large number of patients. I want to make a very important point here. Just like in other repeat disorders, this is a statistical correlation. So for any given repeat size, you can see very significant variation. So here you can see age of onset from 25 all the way to 40. And we have now found even more extreme changes. That tells us that there are other factors that affect age of onset. Part of them are genetic, part of them are environmental, part of them may be instability of the repeat in brain cells themselves. I'd mentioned anticipation, the earlier onset of the disease in subsequent generations, and we now know why that is. In contrast to other mutations, repeat mutations are not stable. They're very often called dynamic mutations. The repeats, on average, tend to expand when they're passed on from parent to children, and you can see this here. Again, this is an affected woman. She passes the repeat on to several of her children. The repeats vary, first of all, 56, 59, 57. And now when we look at her, in the end, grandchild, this means we don't disclose the sex of the individual. The repeat is not 56 or 59. It has expanded to 68. And unfortunately, the age of onset has become earlier. So that is very important when you think about genetic testing, you think of reproductive planning, that these repeats can expand, and then that can mean for you that you may have developed the disease at age 40. It can actually strike earlier in your children. Here's another example from another pedigree. Repeat is 56 and 53, and here it has changed to 57 in the daughter of this individual. And the age of onset is much earlier. So I told you that we thought by genetic analysis is the repeat. It seemed to show that the repeat actually originates in one location, and we engaged in tracing this back by Ancestry. There's a good friend of mine is head or was head of the Marcus Garvey archive at UCLA, and we used to have coffee together. And at one coffee, I told him that geneticists had just identified that Sarah Hemmings likely had children with our third president, Thomas Jefferson, by genetic analysis. And I still remember he put his coffee cup down and smiled and said, "Well, I'm glad that you geneticists are catching up with us historians, because we have known this for a long time." Because whenever Jefferson came back to Monticello, 9 months later, a child was born. So this reminded me when we now traced the genetic history back by ancestry records and helped by many archives, both the archive in the Mormon Church, the LDS Church, by U.S. Census records, by birth records in southern Sweden. Patty and colleagues were able to trace information from our Georgia and Utah pedigrees back to southern Sweden. Here you see some of the records that are available. It's actually quite remarkable. And she found that the Utah pedigree actually traced back to Swedish immigrants who likely were converted to the LDS faith in Sweden, and their descendants then went to the United States and were part of the Mormon trek from the middle of the country in 1847 to the Utah Valley, which at that point was known to exist, was known to have water, but it was really quite an undertaking to decide to settle in a valley that for most of the year is rather dry. And through these records, we were able actually to connect the family that is now in the Utah state of Georgia to the Utah family, to two families that actually lived very close together and had a commercial collaboration. And Patty also found out that looking at shipping records, that a woman was on a boat from Sweden to via Germany. She was with child and traveled to the United States. This just shows you a little bit where these families are from in southern Sweden, and to this point, actually, all these families trace back to that location or to northern Germany. For those of you who follow history, there was a big war in the Middle Ages, 30 Years' War, where Swedish troops came down to what then was the Protestant part of Germany. And so there clearly was a lot of mixture of genes at that time, and in subsequent times when people left Sweden, either settled in Germany or went to the United States. So I think I had shown this to you already. Let's talk about, now that we know the DNA change, what does the disease phenotype, what do the disease manifestations look like? And this summarizes the clinical manifestation in the 8 families that we identified in the United States and in Germany. And it is similar to what other investigators have found in patients with SCA4. So the most important part is really the gait unsteadiness. And I mentioned to you that it's a mixture both of a sensory ataxia, the feedback to the cerebellum doesn't function well, and the problem in the computation of the cerebellum. The neuropathy, the disease of long nerves, that's what neuropathy really means, is related to getting position sense in your fingers and toes and ankle back to the brain. And that shows up mainly when you deprive the brain of visual input. We can actually make up quite well for loss, let's say to some degree, loss of this position sense by visual cues. Why is this practically important? When you walk at night, you go to the bathroom in the middle of the night, don't turn the lights on, you don't have the visual input, and you really have to rely on the input from your toes. And then in SCA4, the gait unsteadiness becomes much worse. And it has very practical consequences. You have to turn on the light at night. We probably should all do it, because actually with advancing age, we all have a touch of neuropathy, even if you don't think so. So that's a piece of practical advice. What is interesting is that some individuals with SCA4 have a chronic cough that is not caused by any consequences or any malfunction in the lungs or the throat. It's what we call a neurogenic cough. It's probably a hyperactive cough reflex mediated in the brainstem. Patients that I've seen with a cough, they either say they have to run out in the cold in the winter here in Utah, or stick their, and I'm not making this up, stick their head in the freezer and the cough stops. So just the opposite of what you see with what is what you call cold-induced coughing, and what you see in asthma sometimes, where coughing gets worse when you are exposed to cold air. It's just the opposite. It makes it stop. What is also important is that there is a dysfunction of the system that regulates our blood pressure when we get up from a lying or sitting position to a standing position. So that's important to know, to remain hydrated, and sometimes to just get up slowly. Make sure that you contract your calves before you get up to avoid having a syncope, a sudden loss of blood pressure. And this is also related to other signs of dysautonomia in terms of constipation or diarrhea, or erectile dysfunction and incontinence. I'd mentioned at the beginning of the talk that actually quite amazingly, in addition to us, 3 other groups identified the SCA4 mutation virtually all at the same time. Fortunately for us, we all found the same mutation. We agreed on the mutation, and really the clinical description of the findings in SCA4 are also virtually identical. So it's very good to know these groups. The other 3 groups are either in Sweden, 2 of them in Sweden, and 1 is located in the UK. So as you all know, MRI is very important in neurological diagnosis. What does the MRI look like in SCA4? Here is a patient, and you can see In contrast to other disorders that are caused by GGC repeats, the white matter, the cables that connect nerve cells don't show any abnormalities. But what you see here, especially, you see a mild cerebellar atrophy, and it's probably hard to see for the non-radiologist. The spinal cord is also too thin, relating to these sensory abnormalities that I had mentioned. In contrast, for example, to SCA2 or SCA1, the brainstem that you see here, this is something we call the pons, is normal in the MRI. So overall, the atrophy change in SCA4 compared to diseases like SCA2 are actually relatively mild. What is different from these other SCAs is the atrophy of the spinal cord. So when you, of course, you had seen the tagline for my department, Department of Neurology, where cures for brain diseases begin. So we, of course, we want to identify genetic mutations with the ultimate goal in mind to treat the disease. So when you find a DNA variant, a mutation in a disease, it is not immediately clear what the mechanism of disease is. Is it that the repeat causes exaggerated gain of function? Does it make this transcription factor work more, but in its normal function? Or does it lead to a toxic function? Does this repeat lead to malformation of the protein and cause disease unrelated to the gene function itself, but just based on the fact that the proteins clump together? Does the repeat lead to a loss of function? For example, does the repeat interfere of transcribing the DNA into RNA? We think that's the cause for diseases like SCA27B, Canvas, and Friedreich's ataxia. Or is it that the mutant protein interferes with the function of the normal protein? We call this dominant negative. Each one, you can imagine, each one of these mechanisms necessitates a different molecular therapy. If it's a loss of function, you want to probably add to this function, maybe by gene therapy or other measures. If it's a gain of function, you want to turn the disease down. I can tell you, it is still a complex question that is not completely answered for many diseases. We had an interesting input into our thinking about the SCA4 gene. Interestingly, also happening at exactly the same time that a group that is interested in neurocognitive disorders identified mutations in this transcription factor ZFHX3, not repeat expansions, but other either changes that make the protein not to be made or with misspecified amino acids, and it leads to a neurocognitive syndrome. What this told us that likely loss of function was not the primary way this repeat acted, because otherwise we would have seen a neurocognitive disease. And we know that actually SCA4 patients prior to onset of their degenerative disease have normal development, normal cognitive function. So we analyzed this also at the molecular level, and I don't want to talk you through this in detail, but we looked at cells from patients with SCA4 with the expanded repeat, and we could see that actually here is the normal protein with a repeat of 22 or 21 repeats, and here you can see a band that tells us that this protein with the expanded repeat is made, so it is present. It is not the fact that the repeat interferes in making the protein, but the protein is made. And we have another part of evidence. We have one patient who died with SCA4 at an advanced age of 70. We obtained his brain at autopsy. Here you can see actually that Purkinje cells are largely gone. And when we look at these Purkinje cells under the microscope in more detail, we see that these cells, these are not Purkinje cells, these are cells in the brainstem. They all have an abnormal aggregation of protein. And we know that this aggregate here in the nucleus of the cell stains with an antibody to this transcription factor, but it also stains with other antibodies that tell us that something is not functioning normally in the waste disposal system um, of the brain of these cells. And that system is called autophagy. It gives us a hint that there's actually something wrong in the way the brain can clear uh, waste, and that happens constantly, and it's important to happen correctly. And we think that to some degree, this expanded polyglycine protein glums up the machinery that deals with normal waste removal. So I'd mentioned that I'd shown you the slide of repeat diseases that cause nervous system disease. And this is just a list of the more common repeat ataxias. Here are the polyglutamine ataxias that were identified for the first time in 1993 with SC1, and then these other common polyglutamine ataxias. Recently, there were new repeat ataxias that were caused by repeat expansions, not in the exon, the protein coding part of a gene, but in the introns in between the exons, a disease called canvas that has actually a pentanucleotide repeat that is made up of 5 DNA bases, SC27B that is made out of a triplet repeat that is actually the same as in Friedreich's ataxia. And we can now add SC4 to a novel category of ataxias called polyglycine diseases. And it's not by itself. There is a relatively rare disease, Fragile X tremor ataxia syndrome, and diseases that are common in Asia that lead to diseases that go along with seizures, cognitive decline, and ataxia, and very rarely also with muscular dystrophy. So, but this SC4 is the first polyglycine ataxia that we know about. Actually, we don't have to look at this. So, when should we think of it? When should a patient with ataxia or a physician think of SC4? And I have to say, this is at the current level of knowledge. I will be embarrassed when tomorrow there's a report that SC4 was discovered in Japan. It's clearly possible. It could be that that patient traces their ancestry back to southern Sweden by a mother, grandmother, or great grandmother. But at the moment, we think that everybody has European ancestry, especially to Northern Europe or Scandinavia. It has this unusual combination of a sensory and a cerebellar ataxia. And really, we define SC4 at the molecular level that we show in a sequencing report that there's an expansion of this GGC repeat to a length of more than 42 repeats. When should you not think of SCA4? If you have an inheritance pattern that is not autosomal dominant, that does not show vertical transmission of the disease from parent to child. I told you that typically the ankle jerk disappears in patients with SC4. So if you have brisk reflexes, all reflexes are present and maybe even exaggerated. It's unlikely that you have SC4. Or if you have other changes in the ZFHX3 gene, for example, changes that incorporate a different, an abnormal or unusual amino acid, that at the current state of knowledge does not mean you have SC4. What to do? And why is it important to find out that if you have SC4? I treat a lot of patients with ataxia and other neurodegenerative diseases, and I find that in most patients, it is important to put a name to your disease. What it also does, it prevents, a lot of unnecessary workup. It also allows you reproductive planning. I think these are important aspects, and hopefully in the future, it will lead to very special therapy. At the moment, we don't have this. So what to do? I had already mentioned this, avoid walking in poorly lit spaces. Falling is the major danger when you have an ataxia. Stay well hydrated so you don't faint especially in the summer. And then something that is very important. It's not the magic bullet, but I tell it to all my patients because it makes a difference. We know this actually makes a big difference in animal models of disease. To stay physically active, as safe as it is, stationary bikes are good, recumbent bikes are good. Stay mentally active, eat healthy, and maintain a good circle of friends and family. And if you look at those 4 pieces of advice, it's probably something we all should follow, and it will make our life happier. So this is the summary. Before I introduce you to the team that led to that helped identify the repeat, SCA4 is defined by finding by genetic sequencing that there is an abnormal GGC repeat. It's an example of a polyglycine disease. The repeat specifies the incorporation of glycine into the protein. The phenotype is more than just the cerebellum. Of importance is autonomic dysfunction, chronic cough, and this mixture of sensory and cerebellar dysfunction. The brain shows inclusion bodies that we think indicate that the waste removal system in the brain is not functioning well. And we have a cellular model of SCA4, and that will be important for us to look at different ways of treating the disease. Here's the team that helped. There was several individuals in my lab. First author is Patty Figueroa, my lab manager, who has worked with a lot of patients also and organized our effort. Sharon did the biochemistry, Mandy the cell biology, and Dan Scholes helped with all aspects of the disease. He's my right hand in the lab. And thanks to all of them. And thank you for listening. And I'm delighted to answer some questions.
Thank you so much, Dr. Pulst. We're going to jump right into questions. Just a reminder for everyone that you can submit questions using the Q&A window at the bottom of your screen. We're going to start off with some questions around SCA4 genetics and genetic testing. So the first question, we have someone who's asking if someone knows they are at risk of developing SCA4, do they need to wait until they are 18 years or older to access genetic testing?
So the answer goes in 2 parts. If they are affected, we can do testing earlier. It's part of a diagnostic workup. If it is a parent who wants to get a child tested, we advise currently against it because we believe in the autonomy of patients and the maturity to make decisions, and so we advise against testing in patients that are not adults. There's a tiny quirk to it in the U.S. legislation. You are an emancipated adult if you get married and if you're younger than 18. And the other exception is once we have a potential therapy for a neurodegenerative disease, then testing would be seen very differently. For example, in cancer syndromes, testing can be done earlier because there may be ways you can diminish the effects of the mutant gene.
And then another genetics related question. Is the transmission of SCA4, is that impacted by the gender of the parent at all? Are you more likely to get it if you're receiving it from your mother or your father,
Great.
anything like that?
Fantastic question. I forgot to point this out. And when you look at the family pedigrees, you can see that the disease is passed on from mothers or fathers to children. It is too early to say if there is a specific tendency of the repeat to expand, especially large expansion, if it comes from the father or the mother. We have seen it in both, and we probably have to study more patients to know. In many of the diseases like SCA2 or Huntington's disease, these larger expansions happen only if the repeat is passed from the father to a child. But then the sex of the child does not make a difference.
Very good to know. And then we have someone who asks, if I have tested positive for SCA4, is it possible to have children without passing on the gene? Could you perhaps talk a little bit about the
Yes.
reproductive healthcare options that could be
Yes.
available?
Yes. So there are several options. Depending on your belief system, um, if you don't believe in having testing of the embryo and or young fetus, and then undergo a therapeutic abortion, you can also do what is called pre-implantation diagnosis. You can use in vitro fertilization, test a part of the embryo with very few cells and see if the mutation is present. That is done now for a number of diseases and provides a very reasonable option um, for reproductive planning. Of course, I think with late onset diseases and diseases where the mutation can get worse when you pass it on, we sometimes see that a parent is diagnosed after they've had their children because the disease started at age 40, at age 50. And that's how these diseases are able to be um, passed on to subsequent generations.
And then moving on, we have some questions around genetic anticipation.
Yeah.
So one attendee asks, you know that there is anticipation in SCA4. What is the most or the average number of generations that you notice before you start having patients experience symptoms at an earlier age?
It can start right from the beginning. I mean, sometimes we as physicians and geneticists, it has taken us some time to realize it because especially in brain diseases, that do not have dynamic mutations, you see quite a range of age of onset caused by different genetic background, by environmental things. So in science, it takes us a way to some generations to recognize it. Now it can happen really in the first generation, and it can not have it really. It's related to the expansion of the repeat, and sometimes the repeat remains stable, and sometimes it expands.
And then we have another attendee who shares a little bit of her husband's family history of the now what they know as SCA4 being passed down and symptoms occurring
Yes.
earlier and earlier. Are there any interventions or therapies that can be done to slow down this progression, either for her husband or for the next
Yeah.
generation?
Yeah. It's so a specific disease-modifying therapy is not available at this point. But what I mentioned, one of the last slides of my talk, staying physically active, physical therapy is marvelous. It actually, in most trials for SCAs, physical therapy has an effect that is greater than that of any intervention. And the other things also staying mentally fit, um, eating healthy, not gaining weight because it is much easier to be coordinated when you're not overweight, and having a great circle of friends. And I cannot overemphasize this a lot. I think we are all very proud human beings. We don't want to be seen staggering into a restaurant. We don't want people at the table think we are drunk because we don't speak very clearly. Um, I tell my patients, you have to get over it. And people, once you tell them, I have a neurodegenerative disease, it's called SCA4. You may never have heard about it. That's what I'm living with, but I enjoy your company. And nobody will ask other questions. Most relatives, friends are very understanding and enjoy the company. And so it's important not to become isolated.
Well, that is all the time we have for today, Dr. Pulse. Thank you so much for joining us and for teaching us more about SCA4. We really appreciate it.
It was a great pleasure. Thank you. And keep the NIH in your mind and contact your representatives. It's important for all of us. Thank you.
Thank you so
Take care.
much. And for all of our attendees, I hope that you enjoy the session and you learned a little bit more about SCA4. Wherever you are in the world, good morning, good afternoon, good evening. Have a wonderful rest of your day and a great weekend ahead. Take care, everyone.